I'm going to talk a little about philosophy, and then we are going to talk about, we're going to move into actually looking at the paper. So keep the paper handy, but let's talk outside the paper for a second. How do you provide context for what a paper is doing? So here I'm going to provide you all the context. I chose only a little bit of this to go on the paper, but as you get your presentations ready, you should be looking for similar kind of context. And when you find the one or two bits of context that are really important to understanding the paper, that's worth putting into your talk. So what this is, and this is one of the ways that I used to start it off. I actually started off talking about how it's a counterfactual. If you've heard of counterfactuals before, it's a whole genre. In fact, now it is a whole genre of entertainment. And so I want you to think about what a counterfactual might be that you've seen in entertainment somewhere. But scientists can do a counterfactual. In a sense, we are saying, what if this FC region was just the only thing in the world and you could choose any place? What if you could choose any place to bind? You didn't have any biological constraints. You just have the chemical challenge of binding. That's not the way it evolved. It evolved in a living system with lots of other constraints for it. But the question is a counterfactual. What if it evolved in a test tube? You know, sort of like the AI protein, you know, with no environment and things like that. So is what if questions are prominent, and I really like them. Some historians sort of look down their nose a little bit at this kind of thing because they seem like parlor games, like who can really know how things change. But I want to give you an example of this. There, one of the early ones is there was Jerusalem was under siege by the Assyrians, and actually the Bible records how they were under a terrible siege. They were like mothers eating children and things like that. It's like it was bad. And Assyria had already destroyed the 10 northern tribes, and those were never heard from again. Those are the 10 lost tribes of Israel. But the invading army at the top of its strength was turned back. The Bible describes this as an angel came in the night and slew most of the army, and then the army got discouraged and turned away. You know, and you know, could that have been a description? You know, of a fast-moving plague of some sort? Is the plague something that an angel could have used? I mean, you have theological questions about that. But whatever happened, it's pretty clear that something like that happened. And the army did turn back. What if the Assyrian army had succeeded? What if Jerusalem was destroyed like the other 10 tribes? And what if we didn't have Jerusalem? And what would the world look like? I mean, that's really, and especially for like a Christian to ask, that's BC, it's before Christ. What would have happened if Jerusalem was destroyed? So there's one about the premature death of Alexander the Great. Alexander the Great died very young, but he had already conquered all the way out to India, right? I think that he, basically got out to western, definitely conquered Persia. And then he died, and then the country fell apart. It was divided into 4 kingdoms and stuff like that. What if he had died before he got out there? What if he had died prematurely, very prematurely? And there's a question of that. You know, how would the world have been different without? And one of the things, this even affects Jerusalem's later history, because Jerusalem was Greek-speaking at the time, because Alexander the Great was Greek and he had conquered Jerusalem. You know, it would have lots of effects. What if Napoleon won at Waterloo? He famously lost, but he maybe could have won. There was a lost order that was found. I believe this was before Antietam. That was it was basically a bit of intelligence that allowed the Northern Army in the Civil War to basically win against the Southern Army when the Southern Army was invading the North. The Southern Army was literally invading the North, but they didn't know where they were going to go. They found orders wrapped in an envelope with cigars, like left behind in an old Confederate camp. And so the Union knew where the Confederate Army was. They went to go get them, and they turned them back from invading the North. What if they hadn't found that order? What if the Confederacy, as a result, had won? And so that's, again, these are kind of who could tell, really? What a taxi driver wrought, this is in the, I believe, the mid-30s or late 30s. Winston Churchill was in New York City. He was crossing the street, and he was hit by a taxi. So this is before World War II, before the Battle of Britain. And Winston Churchill's speeches had a big effect on the country's whole way of getting through that. What if Winston Churchill's speeches hadn't been there because the taxi driver was another inch over and ended up actually killing him rather than simply injuring him? So this is a little bit more about Lee's Lost Order. I'll let you look into that if you're into the history of things. But this was given to General McClellan, and he says, I have all the rebel plans and will catch them in their own trap. He knew that because of this paper, he would be able to defeat the Confederate Army, and he was right. So here's another counterfactual. Do you guys know the Star Trek movies that they do, and they literally have time going back and changing so that they can do the original characters, but do them again. This is not the original Kirk for Spock, and but this is it's like, what if Kirk and Spock? What if history had gone a different way, and then you'd have different Star Trek episodes, and that was their idea. Yeah, you can talk about how they do all that, and they actually play off of varying a lot of other things as well. So that's counterfactuals. I think they're kind of fun to think about. Are there any counterfactuals that you think of when you think of like books, movies, TV shows? That are there counterfactuals that you've come across? Like the Star Trek thing, but different. Retcon is one of the ways you can talk about it, but
I think there's like an actual show called What If on Disney Plus. It's like different scenarios of like the Marvel superheroes.
Oh yeah. That's the What If Marvel. What If is literally a whole counterfactual.
Yeah.
I don't know if the comic came first, but it was a comic series. Yeah, so that's a whole series of counterfactuals. Yeah, they do a lot with that. The other thing I'm thinking of is The Man in the High Castle, is Philip K. Dick's idea of what if the Axis had won World War II, and what would things be like. And then there's the counterfactual, by the way, where you say, what if this had happened? Well, then things would mostly be the same. It might look like this would be a drastic change, but actually things would turn out the same. And a lot of the counterfactuals sort of say that as well. And I just think it's a fascinating question. I don't know, anything else come to mind? I think there are like TV episodes. The What If is a perfect example. I'd forgotten about that. I even watched it and I forgot about it. So. But that's what you want to think about. You want to think about what ifs with this. So let's play the counterfactual with the protein. The Fc region of an antibody will bind a lot of different proteins. The C stands for constant. We're actually going to have some structural immunology discussion about what antibodies look like later. And we're going to be using antibodies in the lab. You're going to be seeing if they bind stuff in May. So the antibody region is always there. A lot of things need to bind antibodies. And a lot of bacteria and other pathogens want to block the things that are binding antibodies. So you have like the guys on your side, the good guys, that are in your immune system that need to bind antibodies. And so they bind the constant region. And then you have bacteria that need to block it. But realize that right here, this is one of those biological constraints, not biochemical constraints. If you have an immune system protein binding the Fc region, and you have a bacteria that wants to block it, of course it's going to bind at the same site. It's going to make a protein that binds at the same site, and because it needs to block the protein, it doesn't mean that the site is inherently bindable, okay? So you realize that this is one of the things that makes the question work. And so what they wanted to do to ask the question is, what if we eliminate all the biology from the picture, just had biochemistry, random set of peptides, find one that binds the Fc region 1, and see where it sticks. Will it bind at some random site because it's a random antibody? Or will it bind at this same site where everything else seems to bind? And if so, how does that interaction work? If it's more bindable, why? And that's the questions that we're asking. And by the way, Delano was actually He was basically my age, and but he was a big name because he wrote Pymol and he gave it to people. After his death, the Pymol was bought by another company, and now you'll see there's another company, and that's when we started to have trouble with having to sign up for a license and stuff like that. You can still do that as a student, and you've done that yourself. But he was a computer programmer. He applied his work to biochemistry, and it was like playing a musical instrument. And he wanted to make masterpieces that would be played by everyone. So that's why Pymol was actually, I know that he probably had some frustrations with Pymol, but imagine how much worse it could be. Honestly, most scientific software is not as easy to use as Pymol is. And that's why people used it. It was free, it was good, and it worked for everyone. So, I would like you to look at this and tell me some things you can tell. There's Warren L. Delano. What can you tell about him from just looking at the author list and the author affiliations? Tell me things that you see in this when you look at that.
Since his name's first, he probably was the head of the research of the paper and stuff like that.
Yeah, and in fact, I would describe him as the hands of the research, the one who is actually doing it. Yeah. So like the head in the practical sense is a really important thing. So that's who you usually put first, and that's why I call it Delano and Wells, by the way. I don't know the other two guys, so I just basically call this Delano and Wells. Do you have something you observe?
So you also put names in order of your age?
A little bit. It's more like what matters more is money, you know, or how much funding you brought in. So James A. Wells, I'm not sure if he was the oldest, but he definitely was old enough to secure grants. And so it will tend to be older because the older scientists are the ones who secure the money. So just like James A. Wells, the end is like the sort of inverse of the bookends are the most important part. And you put the most money-making, sort of the head scientist last. This leads to some issues. Sometimes you have scientists that are put on an author list and they really didn't do anything. You're just putting them on because they were a cool name and they said they liked the paper at a conference or something like that. And so author lists have been abused in the past. But in general, that happens in the middle. At the beginning and end, you're pretty sure this person went first. Sometimes you will see that people share the first credit. That's how important it is. They'll have a little star and they'll say these authors contributed equally. That means that they can present themselves, they're co-first authors. That's how important being first is. Another observation?
Yeah, wouldn't they have the last person be the person who's like kind of in charge of the entire lab, or like an advisor to the first person?
Yeah, well, like my postdoctoral advisor was the last one. So yeah, it's just important to say when we're talking about advisor, that is the exact word that people use. Sometimes you talk about advisor and they're kind of like, you know, Lord of the Rings with Theoden and Grima Wormtongue, who's like whispering in his ear. Grima Wormtongue is an advisor. This is a different kind of advisor. They're more like the king. But yeah, you call them the postdoctoral advisor. They really are the primary responsibility. And the other sign, to whom correspondence should be addressed. Do you notice anything funny about this? SunesisPharma.com. And yet you look at footnote 1, graduate group in biophysics, University of California, and Sunesis. The other thing that you can see right here, it's also in San Francisco, but Genentech. There's a little bit of a thing, there's a mingling between Genentech and Sunesis. Have you heard of Genentech, probably? They're one of the oldest and biggest pharma companies, biotech companies. They basically founded the field of biotech. And James Wells was there. But then, and also you sometimes have sharing where you have a grad student who works both at the company and at the university. Looks like you have something like that going on here. But the other weird thing is that James Wells used to be at Genentech. So he probably started this work, he did it at Genentech. Before the paper came out, he moved to Sunesis. And so Sunesis is where you email him now. But probably most of this was done at Genentech. He had a special deal at Genentech. he actually had freedom to investigate whatever he wanted using Genentech's money. That was really unusual, and yet that still wasn't enough. He still wanted to go somewhere else. I don't know the whole story behind that, but it sounded like he had the dream job where he was given freedom. Maybe he wasn't really given freedom to do whatever he wanted, and maybe there was trouble in paradise. But yeah, that's how this all works. And the other thing about the title is that you all keyed in on the key ideas are in the title. So here's a picture of the FC region of an antibody. Antibodies are Y-shaped, and we'll talk about all the other domains later. The antibody, the part that binds the antigen is the FAB region of the antibody. FC comes after A and B, so you can see it's secondary to binding antigen, but it's also C for constant. And you can see that even though this is a German slide, you can understand there is an FC receptor on granulocytes, which is a kind of immune cell. So this is how an immune cell, if you've ever wondered, we talk about antibodies binding all the time. Well, how does the immune cell bind the antibody? It's because there's a constant region that the immune cell grabs onto, and then it does its thing where it pulls the antigen in and things like that, kills the other cell, whatever. So the other thing is, like I mentioned a few minutes ago, bacteria can actually bind the constant region too. You can find bacterial proteins that bind the FC region. And this makes sense because if the antibody is doing this, if the antibody is binding through this, the bacteria wants to get away from the antibody by blocking this. And so bacteria, they can evolve a lot. They're really masters at making small proteins that bind tightly. And there's even more than one bacterial protein. That's how much they like to make proteins that block the FC region. And they found that these two proteins actually converge. So this is the first sign of convergence. We have convergent evolution among two totally different bacterial proteins, but they both bind the FC region. Again, that's cool, but it's not terribly surprising because they could both be trying to block the same thing. If you're trying to block the same thing, if you're trying to block people from getting into the basket, you're going to hang around the basket. It's that kind of thing about the bacteria. So here's the abstract. This is, oh yeah.
Oh, can I step out real quick? My boss called me.
Okay, sure,
Sorry.
sure. So here's this is the convergent evolution that they found, and this is older biochemistry, but it's how they found what bound in the first place. If you look at the abstract, they found they started with protein G, which was a bacterial cell wall protein that could bind antibodies, and it bound the FC region. They found in this one they found proteins A and proteins H. They didn't have any homology to protein G, but they both bound the FC region and they blocked binding. So it looks like we have three versions of the same idea here. That you have the three that are, it's an unusually clear example of convergent evolution. Three things in red on top, one thing in blue on the bottom, and not to mention the thing in blue on the bottom was also what binds the receptors and things like that. So protein A, protein G, and protein H. The Theodor Geisel protein naming convention. Do you all know who Theodor Geisel is by his more prominent pen name? Give you a hint, he would give names like protein A, protein G, protein H. He's a children's author, and he has it. In fact, the whole book about I think the creature A, thing B, thing C. Ring any bells? You guys read Dr. Seuss? So that's Dr. Seuss's real name. So it just reminds me of thing one and thing two, and there's this is in Cat in the Hat. Cat in the Hat comes back. I believe you have thing A, thing B, and so it's a nice simple. It's a lot simpler than many of the things. And I just got to say that they're too big for these shirts now, but we have 4 boys, and here are their shirts. We had to get them all thing shirts. The problem is they keep growing, and so we knew that we could only take these pictures for like a year or two. We could pass down some of the shirts, but we tried. And there's also a shirt called the Mother of All Things that you can buy from the same store. This is at Universal where they have a Dr. Seuss land, and so you can buy these shirts. Anyways, but it's a nice simple naming convention. Now, that's not really kind of thing that you'll have in your own talk, but I have to show the pictures. They are all bacterial proteins. They all bind the Fc region. And none have any detectable homology. You put the sequences side by side. There is no similarity between them. And they all have totally different domain structures. Maybe they're the case where they have the same like alpha helix, beta strand kind of formation. No, they don't have same secondary structure, tertiary structure. They don't have same anything. Okay. So it's really interesting how. So the question is, how do these three very different things bind in the same place? They're also small, and so you can narrow down. One of the things they like to do is they narrow down this 50 residue domain would bind, and they ran, I think they were doing this with like gels and blots and stuff like that. What they did is they actually could map out the sequence, the secondary sequence. You see you have alpha and beta, you have the turns right here, and you can map that out. There's one alpha helix in the middle there, and this is for protein G. And so what they did is they cut down protein G into different fragments. You cut it down like this, it still binds. You cut it down like that, it still binds. That it still binds. But then you do 5 different fragments, and only this one binds. You line up all the ones that bind, everything with this sequence binds. And so they said to themselves, this amino acid sequence, it's only like 12 residues. This is the center of the binding site. And they were able to find that pretty easily, just with peptide synthesis and things like that. Again, this is a pretty old paper, 1992, right? And so they mapped that out on the structure where that is, what is the most important part for binding, and it's this loop right here between the alpha helix and beta strand 3. So they said, we know that that's the most important part for binding. They can look as hard as they want into the other proteins. There's no other sequence like it. So somehow this works, but there's 2 other sequences at least that also work well as well. So lots of proteins will have a reason for binding the Fc region. Staphylococcal, these are staphylococcal proteins, protein A, G, and H. The Fc receptors will bind on the surface of the immune cells. And there's a Fc receptor that is in the fetus that actually I have that wrong. It's in the newborn because it's neonatal, right? It's newborn. So a neonatal Fc receptor in the newborn. This is located in the stomach. Why would a newborn need an Fc receptor in their stomach of all places? You have to think about a little biology for this one, though. Any ideas?
Something about food from the mother into the stomach.
We're mammals, right? And so the mother's milk actually contains antibodies because the baby can't make antibodies. The baby isn't trained yet. But the mother passes the antibodies and continues to do so after birth through the milk. So you have a receptor for antibodies. It's one of the reasons why infant stomachs are not acidic yet. Because if they were acidic, it would destroy the antibodies. And one of the reasons why you can't give them honey until they're 1 year old or something like that. So anyways, there's this receptor in newborns. There's the Fc receptor on immune cells. We have what that is. And there's another antibody that's called rheumatoid factor that binds this. Now, think about this, rheumatoid factor. This is not necessarily a good thing. What does rheumatoid remind you of? Finish the sentence, rheumatoid blank. Yeah. So arthritis is one case can be caused by your body attacking itself. One of the ways it can attack itself is accidentally making antibodies to it. Antibodies that bind antibodies are bad news. And these collect in the joints, they cause inflammation there, and I believe it's a factor in rheumatoid arthritis. So, but the interesting thing about this is, again, this antibody is something that could bind anywhere. It's an accidental binding event, right? Well, guess what? It binds at the same site. So that was our first clue that maybe there's something special about this site that is binding. And the question is, what would a completely new protein do? Now, one of these things is not like the others. Fc gamma receptor, that actually binds at a different point on the Fc region. It's the odd one out. So we actually aren't going to talk about that. But we're going to talk about all these other things. 1, 2, 3, 4, 5 things that bind. And then here's 6 that bind in the same place. Fc gamma receptor binds in a different place. So here's the protein G and protein A binding to the same site. You can see protein G here, you can see protein A there, and they overlap them there. You can see the secondary structures don't align. It's not like there's a helix that fits into the groove. It doesn't happen either way. And you can even like color the contacts. So here they're green, here they're purple, and but here they just don't really line up. Not even the sites bind up. And this is the thing that we talked about before, about how there was a charged polar interface for protein G. And if you look at the amino acids that are interacting here, they're mostly hydrophobic. So it's not like it's the same kind of chemistry. So no sequence homology, no chemical homology. We can't figure out why they like to bind there. They just do. Fc gamma receptor, by the way, that shows us most of the other ones. See how we're binding between the joint between the 2 C regions? Fc gamma receptor actually binds in the middle of the antibody. It has its reasons for doing that. And so it's a big complex receptor. It's a different category than what we're talking about here. Definitely constrained by biology. So we won't look at it anymore, but we will look at the other 5 or 6 things that bind down below. By the way, this is where Fc gamma receptor binds. It binds in the middle of the Y of the antibody. Totally different binding site, and totally different function. Okay. So almost everything else binds at the same place. Here's the rheumatoid factor, by the way. You see, I'm sorry, this is neonatal Fc receptor. Totally different shapes. And by the way, if you look closely at this, you can see the same antibody domains. This is rheumatoid factor because it has the antibody domains. It's binding the same place. Neonatal, it could bind anywhere, and yet it's binding the same place. So the fact that these 2 bind in the same place is not as surprising, but these 2, that seems weird. And that's why they wanted to look at it. That's why they have the hypothesis that maybe there's something biochemically bindable in this site. And maybe we can make some money for our company by finding out what makes a bindable site in a protein. Okay. So I would like you to do this. I want you to look through the paper now, so please get out your paper. And I believe it's not on the first page, so I'll give you that hint. But I'd like you to look for where they describe designing the library. And I want you to sort of write out or circle what is the peptide that they were designing, where's the program that they used to randomize sequences, and then find the FC1 and FC2 sequences and compare that program to the actual spacing between the cysteines and look for the shared sequence that I told you about. This is where I want you to look at methods. So please go ahead and do that for a few minutes.